So just a quick note about the course. So, of course, you know there is an exam a week from today. And it will be 3.5 questions per lecture hour on average. So 35 questions. There will be some extra credit questions on there. And I think, you know, it'll be your first time to see my questions, but they basically test memory and a bit of comprehension and analysis. So I think they're all pretty simple questions. You know, most of them can be addressed in a minute or so, but a few of them will make you think, and some of them will just test your general knowledge and memory of particular details, kind of a mix of those two types of questions. If you want to, I've already posted all of the lectures up through the end of, or it should be visible to you what the lectures are available until next Monday. So if you look ahead at Monday's questions, I put a couple of example questions on there at the very end of the lecture, but you know, you'll see it's fairly straightforward stuff. So I hope you all do well. And you can contact me if you would like some extra help. It's especially helpful if you have specific questions that you want to address. Just shoot me an email. Okay, and so we'll just go ahead and get into this. So this is going to finish off receptor theories. And the last time we talked about occupancy theory, rate theory, and a few other considerations. Today, we're going to go into some more detail on receptors, largely using growth factor receptors as some more detail. So we're really going to get into how receptors function and what happens downstream of receptors to some extent today. So here are the objectives. We're going to talk a little bit and introduce signal transduction in more detail than you've had before. That's all the stuff that happens downstream from ligand binding to a receptor. And then we're going to examine growth factor receptors, a particular class of receptors, because it offers a great way to show you how signal transduction works. And then we'll go and look at a few drugs that block growth factor receptor signaling. Okay, so first let's talk about receptors, and we're going to get into second messengers and what goes on downstream of the receptor. Okay, so once again, you've seen this slide before. It talks about different types of receptors, GPCRs, ligand activated channels, growth factor receptors, and nuclear receptors. So today, we're going to focus on growth factor receptors. So the other pathways will be discussed in subsequent lectures, many of them for after the midterm. But growth factor receptors are going to be the object of study for today. And what you'll see about these receptors is they have single transmembrane domains. They bind growth factors, not surprising given the name, huh? And they tend to dimerize when they bind their ligands, and so they don't work unless they dimerize. Then they become phosphorylated. In fact, they phosphorylate themselves on the cytoplasmic face, and they also can activate phosphorylation kinase cascades downstream. They can make use of second messengers as well. And ultimately, they activate pathways that result in cell signaling, oftentimes leading to growth and proliferation. Because they're growth factor receptors, what do growth factors do? They oftentimes stimulate cells to divide. So that's the general thing we'll be going through. And now let's get into this business of signal transduction. So, once again, I think you've seen this before, but it's a good place to review. What happens during signal transduction is the primary messenger, which is the ligand, and sometimes we mimic the ligand with a drug. It could be an agonist if it activates the receptor. If it inhibits this receptor, it's an antagonist. Those are the basic subdivisions of the types of action. But that's the primary messenger, the first messenger, if you will. this receptor binds some kind of a ligand. And then a bunch of stuff happens downstream. So the receptor-ligand interaction couples to different intracellular processes, and that's why we call it signal transduction, because the signal mediated by the ligand is transduced. That means it's going to be communicated to downstream steps. Now remember, a given ligand might bind to different receptors on different cells and mediate different intracellular changes. So we're going to see how the type of cell that bears the receptor is going to have an effect on what the outcome is. So for instance, you could have the same receptor expressed on the surface of bone marrow cells as on maybe lung cells or something like that. And you might get different responses because they're different cell types, even though two different cell types have the same receptor. But once again, the first messenger is the extracellular ligand. The second messenger is usually a small molecule that is altered in response to this ligand binding. And we'll go through some of these. But they act kind of like intracellular ligands. They're ligands that go on and influence other molecules within the cell. So in some regards, they're somewhat analogous to the primary messenger, except that the second messengers are generated inside the cell itself and then will interact with other proteins. Another thing that you'll see in the signaling cascades, signal transduction, is that kinase cascades oftentimes lie downstream of the second messenger. And so what is a kinase cascade? Well, a kinase is an enzyme that phosphorylates things, right? So oftentimes you'll have the second messenger causing phosphorylation of a target protein, which is itself a kinase, and that first kinase will then phosphorylate and activate a second kinase, and that kinase will phosphorylate and activate a third kinase, or something like that. But then ultimately, that causes one of two things to occur. You can have cellular responses that occur, and that oftentimes happens in the cytoplasm. Maybe you open or close channels, or you change, the transport of some protein across different organelles. And so you can have kind of very quick early responses, which we'd call cellular responses in this diagram. Or at the end of the kinase cascade, you can cause activation of transcription factors in the nucleus, and then target genes will become transcribed. They'll be expressed as messenger RNAs, and you'll make new proteins. And that's what happens a lot with these growth factor pathways, because it turns on genes that are involved in cell proliferation. Things like cyclins, for instance, which are involved in regulation of the cell cycle. So the cellular response pathways that happen in the cytoplasm tend to be very quick. The stuff that happens in the nucleus tends to be much slower because it takes time to activate messenger RNA expression, and then you have to have translation of those messenger RNAs. And so that's a slower response. Now, both of these things can be going on at the same time. You can have these kind of immediate early cellular responses and subsequent nuclear responses involving transcription. Both those pathways can be active. But here's something important. The cellular responses don't tend to change the protein content in the cell, but the transcriptional responses do, because you're turning on genes, making them be transcribed, they're activated, and you make new proteins because of that. And so the transcriptional responses will actually change the types of proteins that are in the cell. So let's talk now a bit about these second messengers before we get into how they actually work. But basically, there are a handful of second messengers that have been identified in cells, and they tend to be very important molecules, but they can be divided and classified kind of on their physical attributes. There are certain second messengers that are hydrophobic molecules, and that means that they don't freely dissolve in the aqueous environment of the cytosol. And oftentimes they tend to be, the reason they're called hydrophobic is they have these long tails that can insert into membranes. So a lot of these hydrophobic molecules have tails on them, amino acid sequences that are hydrophobic themselves, and so they can insert into the plasma membrane, for instance. And they can diffuse around in the plasma membrane, but the interior of them is inside the cell, okay? Secondly, we have, and examples are diacylglycerols and phosphatidyl inositols. So here's diacylglycerol, here's phosphatidyl inositol. This one's actually called PIP3 because it's phosphatidyl inositol with 3 phosphate groups added to it, okay? But you'll notice that PIP3 and diacylglycerol have these long kind of tails, aliphatic tails. These are hydrophobic amino acid sequences, and they can, they're tethered inside the cytoplasmic membrane. And they will cause other proteins to become activated when the proteins encounter these second messengers. Secondly, we have hydrophilic molecules, and this one here is kind of famous, cyclic AMP. It's adenosine monophosphate, so this is adenine ribose sugar, but you'll notice that the phosphate has an unusual linkage. It's cyclic. It's connected to the 5' carbon and the 3' carbon. And so this is indeed a second messenger, and it's cytoplasmic, and it can actually diffuse around in the cytosol, the cytoplasm of the cell. Here is IP3, and you can see it looks somewhat like PIP3, but it doesn't have anything that would anchor it into the membrane. So this is actually a soluble second messenger that can also trigger responses. So these are all hydrophilic. Like I said, some examples include cyclic AMP, IP3. Even calcium can have some second messenger features, okay? And calcium ions are quite soluble. And in addition to cyclic AMP, there are some pathways that use cyclic GMP, guanosine monophosphate, okay? And lastly, we have some second messengers that are actually gases. And these would include, for instance, nitric oxide, carbon dioxide, especially nitric oxide. And so these can be in the membrane or the cytoplasm, and they can trigger responses in cells as well. Okay, so those are the kind of physical and solubility attributes of these, but all second messengers can be classified into one of these 3 categories. So let's look at one of the most famous ones, cyclic AMP. That's a second messenger, and let's look at what it does in a pathway. And here, there's already, the receptor is not shown, it's upstream somewhere. But once cyclic AMP is generated in the cell, it binds to a protein called pKa, protein kinase A. And it activates pKa, and pKa, so the regulatory subunits bind the second messenger, cyclic AMP. The catalytic subunits of pKa are actually kinases, and they, can phosphorylate themselves, and indeed leads to a series of phosphorylation events, a phosphorylation cascade, which is called a kinase cascade. But at the end of the day, the last phosphorylated kinase phosphorylates a transcription factor called CREB. And CREB is a very important transcription factor, and it activates many different target genes. So one of the features of these signal transduction cascades that end up activating transcription factors is that you don't oftentimes have just one transcriptor that's activated, but you might activate 5 or 6 or 7 different transcription factors. And each of these transcription factors can regulate a different, larger number of genes. So you can activate many genes just because of one signaling pathway. Okay. So I just want to back up for a moment and just keep in mind that even though all of this is going on, ligands themselves can be selective or non-selective. So you can have selective ligands, which could be, for instance, a selective drug. And so here's one that activates receptor 1, and then it turns on all that downstream signaling, and you get a desired effect. You could have other drugs that also activate receptor 1, but maybe these are non-selective, and they activate other receptors, unintended targets, receptors 2, 3, and 4. And perhaps what happens downstream of these are side effects, okay? Unwanted cellular responses. And so, although these drugs might treat the disease, they do so at the expense of activating unwanted receptors that cause side effects. So the selective drug is typically is superior to the non-selective drug. So bear that in mind. We kind of covered that before. Okay. All right. So that's kind of general stuff on signaling pathways. But now let's get into growth factor receptors and use them kind of as a case study of what actually goes on in some real-life signaling pathways. And once again, we're talking about this class of receptors here, the growth factor receptors. Okay. So ligand binding to the growth factor receptors triggers several responses. So when the ligand binds these receptors, ultimately what's going to happen is that it's going to activate cell proliferation, and that means cells will divide. You'll get more of them, right? So that's, if you think about the biological process of growth, it usually means making more cells, okay? But in addition to that, a lot of that growth, which is the outcome, the cellular outcome, it involves changes in gene expression. And so these are typically the target genes or genes that are involved in proliferation. So first you get the gene expression and then you get proliferation. There's another aspect of growth of cells that's very important, and that's cell migration. So cells, when you get new cells, they have to end up in the right place to make a well-defined organ. And so a lot of some of the target genes of growth factor receptors also regulate cell migration, the ability of the cells to actually move around and become motile and change their location. Some of these genes actually control interactions between different cells, so which cells they will kind of stick to and associate with. So lots going on here. But if you look at the molecular biology of all this, how it works, you have growth factors, and these could even be endogenous growth factors, or it could be drugs that mimic endogenous growth factors. And what you'll see is the following. This is an unliganded situation here, where you have growth factor receptors that have not yet bound ligand. And remember, these things dimerize, so these have not yet dimerized. And then you have all of these factors inside the cell that are there waiting to participate in a signal transduction pathway. But they too have not been activated. So the key is that when a growth factor receptor binds ligands, and oftentimes a ligand can bind to each of the growth factor receptors, the same ones, this actually causes dimerization of 2 growth factor receptors into a dimer. And that's kind of a very key regulatory change. And it's caused and requires ligand binding. So when two growth factor receptors dimerize, mediated by ligand binding, some interesting things happen. One thing is that you get autophosphorylation. I'll show that in some more detail in a moment. But then this is now an activated receptor, a dimer actually, and it's going to then kick off signaling cascades that lead to things like cell proliferation, cell migration, and a lot of these actually depend on gene expression as well. Okay. So, one important principle here is that ligand binding to growth factor receptors causes dimerization. That's just how they work. That is one key feature. But another key feature, once dimerization occurs, growth factor receptors, the cytoplasmic portion of the growth factor receptor can undergo phosphorylation. So once again, let's just look at the structure of these growth factor receptors. Here's a dimerized growth factor receptor. Each one of these is a protein that has an extracellular face, and this contains the ligand binding pocket. And then it has a single transmembrane domain. So that's filled with hydrophobic amino acid residues. Oftentimes they form an alpha helix that spans a membrane. And then growth factor receptors have a cytoplasmic domain that's kind of extrudes into the cytoplasm. And within that cytoplasmic domain are multiple tyrosine amino acid residues, okay? They can be phosphorylated. In fact, interestingly enough, growth factor receptors typically execute autophosphorylation. So it's a kinase that can phosphorylate itself, okay? And where does the phosphate come from? It comes from ATP. So ATP contributes a phosphate, and in so doing, it's converted into ADP. So the phosphates come from the source of ATP. Now, interestingly enough, this is now an activated growth factor receptor dimer. It's phosphorylated itself, but these phosphorylated tyrosine amino acids have a very special feature. It creates basically a docking site for other proteins, activator proteins. And these are proteins that have the ability to bind and associate with phosphorylated growth factor receptors, but they will not bind to unphosphorylated growth factor receptors, okay? So that phosphorylation site creates a docking site for other proteins, and then these are basically the first non-receptor components. These activated proteins are the first non-receptor components that then go on and participate in a cell signaling pathway, okay? And so the phosphorylation is a little switch that allows the activated proteins to associate, and then they themselves become active. And then, you know, these arrows indicate a lot of stuff happens downstream from there. So here's a real detailed example of this, and we'll use epidermal growth factor, EGF. So here's the unliganded receptor, the inactive tyrosine kinase activity of the C-terminal or the cytoplasmic region. And it takes two EGF growth factors to get this thing activated. One binds to one receptor, one to another, and that causes dimerization to occur. And this then causes phosphorylation of the cytoplasmic domains, okay? And so there are then these other proteins called adapter proteins. These are involved in the signaling cascade. And one of them is called GREB2, but it is a class of proteins called an SH2 protein. So SH2 proteins are kind of famous. I want you to know what SH2 proteins are. And they are proteins that have this ability to associate with the cytoplasmic domain of growth factor receptors, but only when they're phosphorylated, okay? And so GRAB2 is an SH2 protein that only binds this when phosphorylation is present on the cytoplasmic domain of the growth factor receptor. What happens then? Well, this can then activate another protein called RAS. And RAS is actually, if it becomes mutated, it becomes an oncoprotein, actually. But under normal growth considerations in normal cells, RAS then can activate another protein called RAF. Then that phosphorylates a protein called MEK, which phosphorylates another protein called MAP kinase, MAPK, and then MAPK can turn on and activate transcription factors. And a lot of this sequential phosphorylation can happen on a scaffold protein, so it's all kind of held together in one place in the cell. And that increases the rate of this kinase cascade because it physically sequesters it all into one region. But that's a very typical growth factor pathway, and it's your first real look at a complete signal transduction cascade. Ligand binding, dimerization, autophosphorylation, binding of an SH2 protein, activation of another protein, and then a kinase cascade, ultimately leading to activation of a set of transcription factors. And these transcription factors regulate things like cell proliferation, for instance. Okay? Incidentally, the reason RAS is a proto-oncoprotein is that when the RAS gene is mutated, it can turn on everything here downstream, and it doesn't even need the receptor anymore. It becomes hyperactive, and you can get tons of cell proliferation, and that's what cancer really is, out of control cell proliferation. Okay, so yeah, RAS can become an oncoprotein if the RAS gene is mutated. So receptor tyrosine kinases, once again, just kind of summary, they contain a single hydrophobic transmembrane region. And you know, it's 25 to 38 amino acids, something like this. An extracellular region happens to be the N-terminus of the protein, N-terminal region, and that's where the ligand binds. And the structure of that N-terminal region, a little bit like immunoglobulins, like antibodies, and some of them look like epidermal growth factor. So many different growth factor receptors have one or these other types of extracellular N-terminal regions because they bear resemblance to immunoglobulins or EGF, which is kind of a case type growth factor. You know, they contain fibronectin 3 repeats. That's not particularly important. Cysteine-rich regions. But the intracellular C-terminal region, that's catalytic, highly conserved. And what does it do? It catalyzes autophosphorylation of itself on tyrosine residues. And sometimes it can phosphorylate other things too. Okay, so a little bit of complexity here. Here are 4 different growth factor receptors. IGF-1, EGF, we just talked about that, PDGF, and TGF-beta. These are 4 different independent growth factor receptors. They all have to dimerize and do autophosphorylation, interact with SH2 proteins, all that sort of stuff. You know, their activity is that they phosphorylate tyrosine residues, but they all function in the same general way, except that they bind different ligands, right? What happens downstream from each of these growth factor receptors? And so this is looking at what happens to all of them. Well, EGF can activate RAS, but so can PDGF, that's platelet-derived growth factor receptor, which then activates RAF and MEK, and you get transcription of proliferation genes. These ones here you have IGF-1 and EGF can activate a different protein, PI3 kinase, which activates another kinase called AKT, and that can suppress apoptosis and also, I should say, cause proliferation. Whereas TGF-beta activates another signaling pathway in calling proteins called SMADs, which then exclusively are involved in transcription of proliferation genes. And some of these responses are also angiogenesis, that's the formation of blood cells. I'm sorry, not blood cells, but the formation of blood vessels, excuse me, and even metastasis, which is important in cancer. So, there's complexity that goes on within the cell, and there's also crosstalk and branching pathways. So for instance, if you want to suppress apoptosis, ligand binding to IGF-1, EGF, and PDGF can do that because you follow the arrows and they end up at suppression of apoptosis. But TGF-beta won't do that. So there are three ways to get suppression of apoptosis. One way to get purely transcriptional responses. But you can see that the important point is that all of these internal cellular components here, things like RAS, RAF, AKT, etc., they can respond to different receptors, right? So it's a branching pathway. And that means that there's a lot going on. And I'd just like you to know that because that's how our cells are wired up. But then also think we made a big deal out of drug selectivity, right? Well, drug selectivity can be a problem because of how cells are wired up, because you have multiple ways to achieve the same outcome because of interconnected signaling pathways inside cells. So it's a complex world, and this is kind of what we're dealing with. And scientists have kind of figured all of these pathways out. But you know, if you're designing a new drug, say you wanted to design a new drug that didn't mess with apoptosis but caused proliferation, which receptor would you be working on? Well, probably TGF-beta, right? Because it has this unique pathway. So knowledge of the biology is really important in drug development. It's also important in knowing how drugs work. So there are a lot of growth factor receptor families. There's the EGFR family, and there are several EGFR receptors, epidermal growth factor receptors. One of them is called HER1, which is famous in breast cancer. There are FGFRs, fibroblast growth factor receptors. Another one called VEGFR. This is kind of a famous one for regulating vascular endothelial cells. So this one causes vascularization. IGFR, insulin-like growth factor receptors. VEGFR is interesting because solid tumors need to become vascularized in order to grow. They need a supply of blood. So there's been a lot of effort in trying to inhibit vascularization of solid tumors, because if you can inhibit that, then the tumor can't grow well because it's not getting a blood supply. Okay, so let's now talk a little bit more about EGFRs. These are epidermal growth factor receptors and some drugs that target them. But before I do this, once again, I want to just, obviously I'm not going to hand you all blue books during the exam and say, what is the crosstalk in the EGFR pathways? Because look, this is complex. And I throw this up here, this slide, just to show you how complex all this stuff is in real life. Here's EGFR here. Here's HER2, a whole bunch of receptors. Here are the different endogenous ligands that can bind these various receptors. And then down in this region are all of the signaling pathways. So this denotes the plasma membrane. The receptors are in the plasma membrane, external ligands. And then all of this is signaling that goes on in the cytoplasm, mostly a bunch of phosphorylation cascades. So most of these are kinases. And then ultimately, some of these pathways lead to the activation of transcription factors in the nucleus, and it regulates things like proliferation, survival of cells, angiogenesis, formation of blood vessels, cell-cell adhesion, migration and invasion, and differentiation of cells. And so these cellular responses, turns out, those are all very important in cancer. And what cancer does is it kind of hyperactivates pathways that are usually involved in just cell proliferation. But even without cancer, we need cell proliferation. If you get a cut on your skin, you want that to heal, right? So cells are going to have to divide appropriately and under control to heal the skin. The lining of your intestines is highly proliferative. The half-life of a cell in your intestines is something like 60 days or something like that. And so there's normal loss of cells in the intestine. Those have to be replaced with new normal cells in the right context. And so that's what these pathways are doing in a healthy individual. But when some of these kinases are mutated, you can get cancer, and then you can get hyperproliferation of those same tissues, and you get tumors. But remember, in terms of pharmacology, just because you have a specific drug, a selective drug that activates one receptor, wow, you can hit a lot of different pathways. And other drugs that activate different receptors can activate the same pathways. So that's what I mean by crosstalk. And so this kind of illustrates the complexity of all of that goes on inside the cell. I just want you to appreciate that phenomenon, that's all. So EGF signaling activates many signaling pathways. Here are some of them. You don't have to remember the names of these in particular unless we go through a particular slide that I show you in detail. But these are just the names of different signaling pathways. And basically, you get information in here about what participates in it. You know, phospholipase C, IP3, that's a second messenger, and calcium is a second messenger. INFAT is involved in regulating transcription, for instance. But ultimately, these are the types of responses you get in cells with growth factor receptor signaling, okay? So we'll talk about other receptor classes later in the course that don't have anything to do with these cellular responses, okay? But growth factor receptors are kind of famous in doing stuff having to do with cell growth, division, angiogenesis, cell-cell adhesion, that kind of thing. So here are some drugs that bind to different growth factor receptors. And in this case, I'm showing you a lot of drugs that are actually antibodies. And so, for instance, cetuximab with the brand name Erbitux is an EGFR inhibitor, okay? And these are inhibitors of growth factor receptors, and they're all antibodies. You'll notice that the name ends in MAB. MAB stands for monoclonal antibody. And so humans are very good at making antibodies that bind specifically to individual proteins. And there are a lot of drugs that are actually monoclonal antibodies, and these antibodies inhibit different growth factor receptors. Here's one that inhibits VEGF, for instance. So that's going to be good for some cancers, even macular degeneration, because there's some angiogenesis concerns there, I think. Here's another one that binds IL-2 receptor. IL-2 receptor is also a growth factor receptor, a treatment for MS, multiple sclerosis. Here's Herceptin, trastuzumab, another monoclonal antibody that binds to the HER2 receptor, and that's a growth factor receptor that drives the growth of breast cancer. So you inhibit the receptor, and you can inhibit the growth of breast cancer. So these are so-called biologic drugs because antibodies are actually biological factors. So they're commonly referred to as biologic drugs, but they are monoclonal antibodies or portions of monoclonal antibodies, and they basically bind to the receptor and inhibit it. They also have to be injected because they're proteins. Antibodies are proteins. So if you put an antibody in a pill and swallowed it into your stomach, you're going to digest it. You know, your stomach thinks it's hamburger. It's just going to digest protein. So you have to inject these because they can't successfully make it through your digestive tract. But yeah, a lot of antibody innovation, biologic drug innovation has been focused on growth factor receptors. Here's what this thing looks like. Let's look at cetuximab. And you can see that here is the extracellular ligand binding region of EGFR, a growth factor receptor. And you can see that cetuximab, it's a portion of an antibody called the fragment antigen binding, and it has been designed so it specifically binds to EGFR. Okay? And it's an antagonist. Okay? And so whenever, if you have EGFR that is associated with cetuximab, it inhibits the dimerization of that growth factor receptor. And growth factor receptors have to dimerize in order to signal, right? And so kind of a knock-on effect of that is if you inhibit dimerization, you're also going to inhibit autophosphorylation, okay? So this antibody here has been specifically designed to inhibit dimerization of a particular growth factor receptor. There are also small molecule EGFR inhibitors, so these are not biologics. And here are a couple of them, gefitinib, erlotinib, and you can see that they have some structures that have a lot of, aromatic rings in them, but they are actually inhibiting ATP or mimicking ATP to some degree. They're reversible inhibitors, and they're competitive with respect to ATP. So you'll recall that these growth factor receptors can do autophosphorylation. Well, they need ATP because that's the source of phosphates, right, to do autophosphorylation. These drugs inhibit autophosphorylation in the cytoplasmic autophosphorylation regions of growth factor receptors, in this case, EGFR. Okay. So, this is kind of an interesting thing, and this is looking at the effects of the drug on the Ki of ATP. So Ki, remember, is a little bit like Kd. It's basically a measure of the affinity of the receptor for ATP, and that's going to be involved in phosphorylation, right? And so here is a normal EGFR receptor, and I think we can interpret this. The Ki is 13. Remember, this is kind of the negative log. So the higher the number, the more strongly the binding. So 13 nanomolar. You can think of it almost like a Kd. It'd be, And this is basically the sort of curves you get with ATP only. But if you actually add erlotinib, which is this drug that looks like ATP, it's an inhibitor, you actually change the Ki to 3 nanomolar for ATP. And that's because of competitive inhibition. So what you're really doing with this is competitive inhibition, and the presence of erlotinib is basically preventing or altering the affinity of the C-terminal domain of this receptor for ATP itself, okay? And so, interesting stuff. But, you can do biochemistry on this stuff too. Last thing we'll mention here are VEGF receptors and their pathways. So that's the vascular endothelial growth factor receptor. And so VEGF is involved in angiogenesis, which is, angiogenesis is the process by which tissues become infiltrated with blood vessels. And VEGF is also involved in tissue rejection pathways, interestingly enough. So some transplant considerations. But here is how this works. So here's VEGF receptor. It binds to the ligand VEGF, and that activates, or it mobilizes phospholipase C and causes formation of IP3. So IP3 is one of these second messengers, actually. And IP3, here's what it looks like. Remember, this is one of these soluble second messengers. IP3 then can diffuse, and it causes a calcium channel to open. Okay? So calcium ions then can rush into the cell, and some of it's actually mobilized from internal intracellular stores. But at any rate, IP3 causes increases in calcium levels. And then that actually has an interesting thing. That itself can stimulate transcription of INFAT, but there's also some other stuff going on. It turns out that INFAT is a transcription factor, but it's a different type of transcription factor. It exists in the cytoplasm, but it can translocate into the nucleus. And when it's in the nucleus, INFAT, transcription factor, causes transcription of some cell proliferation genes, but also importantly, some angiogenesis genes. And so this is kind of interesting because INFAT is ordinarily phosphorylated, but calcineurin, which is activated by calcium ions, causes INFAT to become dephosphorylated. And when that happens, INFAT is then dissociates from calcineurin, and unphosphorylated INFAT is then competent to translocate into the nucleus where it drives expression of angiogenesis genes. So a lot going on, but it's an interesting pathway because it shows you a couple of features, the importance of a second messenger system, and also the importance of phosphorylation. So phosphorylation acts like a little switch on most proteins in cells. Sometimes phosphorylation, most of the time I would say phosphorylation actually turns or activates proteins, but sometimes dephosphorylation is activating, and this is an example of that. Okay, so there's a lot that's not shown on this diagram. Like, I would look at it as a scientist and say, why is it that dephosphorylated INFAT doesn't go into the nucleus? Well, phosphorylated INFAT probably has a connection with calcineurin, and there might be transport pathways that, phosphorylation inhibits it from transport. I'm not sure of that, but, that's phosphorylation groups act as interesting little molecular switches, on-off switches in cells. And sometimes phosphorylation events are activating, especially in kinase cascades, and sometimes at the end stage molecule, it can actually removal of phosphate can be important. You also saw phosphate groups on the growth factor receptor C terminal itself. That's a binding site for SH2s. So once again, it's like a little switch. If the phosphate's there, an SH2 protein can associate. If it's not, it can't. So they're cool little molecular switches, and they mediate protein-protein interactions that are phosphorylation dependent. Okay, so a little bit more about this pathway, and a little bit having to do with tissue rejection. So cyclosporin is an immunosuppressant, and it looks like this. It's called cyclo because it's a big long circle, right? It's used in organ transplantation. It's also used in psoriasis and asthma, another condition called dry eye, which are also kind of immune type responses. But what this drug does is it prevents dephosphorylation of INFAT. And if you prevent dephosphorylation of INFAT, you don't have those transcriptional responses downstream. And so, there's a lot of stuff going on here. One of the other things is that some of those genes are involved in T cell activation, and the T cell activation is an immune function. So I guess in addition to cell proliferation and angiogenesis, we could also add in that T cell activation genes are also targets of INFAT. And INFAT is just a transcription factor. Okay, so I think that's it. The takeaway messages here are that you've seen kind of getting a feel for what these internal signal transduction cascades are like. You know that the cell signaling stuff is very complex and interconnected. And growth factor receptors, you've got some familiarity with how they work. And some knowledge of the types of genes that get turned on by growth factor receptor signaling. And yeah, so we'll leave it right there. Okay, I will be sure to post this recording today, and I'll post all of my recordings fairly quickly as time goes on because I know the midterm is approaching. All right, thanks everybody.
Mm-hmm.